Thank you all for joining us today. I'm Dr. Camila Barnes. I earned my master's and doctorate degrees, DNP, from the University of Rochester School of Nursing. I am also a family nurse practitioner, clinician, FNP director, educator, and business owner. I am also a member of the Alumni Board, a group made of 25 members representing each of the university schools with the purpose of engaging all alumni and students in a lifelong connection with one another within the university as well. Today's session is part of a series launched in April 2020 called Experience Rochester that exemplifies the university's commitment to lifelong learning. The series features various topics and speakers unique to the university. Today, we are joined by a panel of URMC neurologists for a discussion on advancements in treating neurological diseases using gene therapy and the optimistic future for many similar conditions. Dr. Emma Ciafaloni is a professor of neurology, pediatrics, and obstetrics and gynecology, associate chair of research in neurology, and director of the URMC Pediatric Neuromuscular Medicine Program, including the multidisciplinary spinal muscular dystrophy, SMA, and Duchenne muscular dystrophy, also known as DMD clinics, and the newborn screen SMA program. Dr. Ciafaloni has devoted her 25-year career to the diagnosis and treatment of adults and children with neuromuscular diseases, particularly children with muscular dystrophies, SMA, DMD, and myasthenia gravis. She has been leading and providing early access to novel treatments for DMD and SMA, including gene therapies. Her research interests have focused on experimental therapeutics and neuromuscular disease, and she has also been involved in many clinical trials in pediatric and adult neuromuscular diseases, including an ongoing gene therapy trial for DMD. Dr. Bo Lee is a board-certified child neurologist and has additionally completed a fellowship in neurogenetics. She directs a newborn screen Pompe disease clinic, a part of the Pediatric Neuromuscular Medicine Program. She sees children with a wide range of genetic neuromuscular conditions, including DMD, SMA, Pompe disease, and other congenital myopathies and dystrophies. She works closely with the newborn screen SMA program and performs intrathecal, which is via lumbar puncture, administration of nusinersen as part of the comprehensive care options for patients with SMA. Her clinical research interests are in genetic therapies for rare neurological disorders. She is a lead investigator of several rare diseases registries, also with Pompe disease, Fabry disease, and SMA, and also the lead and co-investigator for clinical trials in various neuromuscular disorders, including congenital myotonic dystrophy and DMD. And our third panelist is Dr. Samuel McKenzie, who joined URMC in July 2021. After completing his residency at the University of Michigan and a fellowship in neuromuscular medicine at the Ohio State University and Nationwide Children's Hospital. Dr. McKenzie, who is board certified in neurology with a special qualification in child neurology, treats children and young adults with a range of neuromuscular conditions, including DMD, SMA, Charcot-Marie-Tooth, myasthenia gravis, myotonic dystrophy, and various congenital muscular dystrophies. He is a director of the limb-girdle muscular dystrophy program at URMC as well. Dr. McKenzie's research enterprise is primarily focused on developing gene-targeted therapies, including adeno-associated virus, mediated gene replacement, and antisense oligonucleotide therapies for patients with neuromuscular diseases. Other active laboratory projects are focused on developing novel animal models of myotonic dystrophy that recapitulate the somatic instability of cardiotachography repeat tracks seen in humans and improving our collective understanding of mechanisms underlying rhabdomyolysis, which is a life-threatening condition caused by muscle cell breakdown. At the end of our presentation, the presenters will have answers to many of your questions from the audience. As time will allow, you can put your questions in the Q&A box on your screen at any time. Now I will turn it over to our presenters, Dr. Ciafaloni, Dr. Lee, and Dr. McKenzie. Please welcome them.
Excellent. So welcome, everybody. We're very excited to talk to you about this very exciting topic. And, I want to start with a little bit of a history. And at the time of this famous editorial by Marshall Nirenberg, who received the Nobel Prize for Medicine in 1968, at that time, I was 3 years old. So to think that now I've been actually treating babies with spinal muscular atrophy, Duchenne muscular dystrophy, and many other for many years, to actually treat these incurable diseases for the past 5 years is incredibly exciting. And what Nirenberg so ingeniously realized, that he predicted success for translating the knowledge of biochemical genetics into actual human treatment, but he predicted also that there will be a need to overcome formidable technical problems. His prediction at that time was about 25 years before, his cracking the code of RNA into protein will lead to actual human treatment. It took about 50 years, but what's also very interesting about this short essay is that he really predicted also the ethical issues that will come with such a powerful treatment strategy as genetic gene-based treatment, especially when translated into society and system that are very complex and imperfect. Unlike the more controlled environment of basic science laboratory and experiments that are carried in a dish or in an animal model. So it was very much ahead of its time in that sense. And, historically, the field of gene therapy has really lived through ups and downs of over optimism at the time of the completion of the Genome Project, which was finished in 2003. At that point, all the 20 to 25,000 genes were all been discovered and completed. That over optimism was unfortunately followed by some years where, translating into clinical trials had some major setback. The death of Jess Gelsinger was very much one of such step back in the field of gene therapy. This was a young man, 18 years old, with a very rare disease that was infused with gene therapy and died in 4 days from a massive autoimmune inflammatory response. And that's really stalled the field of clinical trials in gene therapy for several years, together with the experience of late onset leukemia in kids that were actually successfully treated for SCID, for an immune deficiency genetic disease, but then due to insertional mutagenesis, they realized that these open up complication later with leukemia. But then eventually the field went back to the bench and really did an incredible amount of work to really solve some of this problem, right? Insertional mutagenesis, better vectors, how to deal with immune and inflammatory responses, really, build on the initial milestones and failures. And you can see here that now by 2017 on, the field has pretty much exploded. And I would say that a major milestone, certainly in neurology, has been in 2019 with the FDA approval of in vivo gene therapy for spinal muscular atrophy, which we're going to hear a little bit more in details what that really represented for the field. And it's important to remember that although the biggest proportion of diseases that are really addressed by gene therapy clinical trials, if you look in this, is really a large portion is really cancer diseases. So you all have heard about the advances with CAR-T and in cancer disease. If you see in this table, monogenic diseases and neurological diseases, so genetic diseases and our neurological diseases really is the second number, but really in much smaller percentage. And yet, because of the perfect storm of what happened with the spinal muscular atrophy, we have really been leading the transformative changes of having FDA approved gene therapy now for human diseases. Obviously, from when the very first patient in 1990, 4 years old, Ashanti Da Silva, was actually treated at NIH. That was the first patient that ever entered a clinical trial for gene therapy. It was successful. She lived well into adulthood with a significant improvement in her disease. Since then, there has been, you can see the numbers have grown exponentially in terms of human clinical trials for gene therapy. And just as to put in perspective, just in 2022, there were 5 FDA approved gene therapy. But really, it has been 30 years since that first patient was treated. And you can see that the number has been growing, and there has also been a good improvement and movement towards more advanced clinical trials phases. So not just first in human and phase 1 and 2, but really getting them to the finish line of more advanced clinical trials. So, and this is just to clarify, because it can be sometimes quite confusing when you hear about gene therapy. So there are gene-based therapies would really address messenger RNA, DNA, and other things. But when we really think about gene therapies, we think of 2 main approaches. One is ex vivo, right? And the cancer field has really led that with the CAR T cells drugs. So where you harvest T cells from the patients, you put them in a dish in a lab, you manipulate it genetically to create chimeric antigen receptors, T cells, and then you put them back into the patient. That's what we call ex vivo transduction. There are several very successful drugs in cancer. But when it comes to neurology and genetic diseases, we're really talking about infusing a vector with a gene through an IV. It's a one-time infusion and is injected directly into the patients. And so the very first, systemic disease, genetic disease, was actually for an eye disease, for genetic retinal disease. So Luxturna was the first approved gene therapy. But then neurology really kind of took the lead with spinal muscular atrophy, approved in 2019. And very excitingly, as June 22nd of this year, 2023, we now have the first gene therapy that has been approved for Duchenne muscular dystrophy. And we're going to talk a little bit more about that. And the basic is really that the majority of gene therapies that are now in trials, they use a viral vector So it's really a capsid. So it's a viral that is usually is not a human pathogen, so it doesn't cause disease in human. It got completely emptied from the viral DNA. And in this capsid, in this vector, we introduce, a gene or a engineered gene. And this is targeted at specific tissue, depending on what the disease that we treat. The capsid then disintegrates in the nucleus of tissues and the cells of different tissues. And really the transgenic material stays in the nucleus and continues to produce and transcribe the RNA and the proteins missed for that particular disease. And that's, obviously is kind of the general recipe that then is, it depends from what tissue and what disease, all different factors. And this is when we look at a drug, a gene therapy drug, really this again is kind of, a vector. There are many different AAV, adeno-associated vector are the most popular right now for genetic disease, certainly in neurology. They are chosen based on a variety of qualities, and usually in the population there is a very low pre-existent immunity towards this vector, so they make it really good candidates. But they do have size limitation of how much genetic information we can put in this vector. They're also chosen depending what tissue you want to address. And then, you have a promoted and allowed the genetic material put into the vector to really transcribe the transgene, for as long as possible. And usually the field has really moved away from we call this a construct or a cassette, to not integrate actually in the main DNA, in the nuclear DNA, because of this early lesson about insertional mutagenesis and causing cancer down the road. So there's been a lot of really improvement in this basic scheme of how you build a gene therapy for different diseases. And, what we are really proud about Rochester, and I know that I'm speaking to Rochester alumni, is that the Rochester way, the neurology department was really a pioneer in patient-centered neurological care and experimental therapeutics. So our department was funded in 1966 by giants of neurology. Dr. Joint and Dr. Briggs very early on realized that what patient-centered care really means is the best possible clinical care, in conjunction though with really the hope of new treatments. And especially, neurology, with the vast number of untreatable diseases, they very early on develop an infrastructure for really clinical research. And early on, they really said that discovery from basic and translational research needs to be tested in clinical trials to really make a difference. So everything starts from the patient, we learn from the patient, we care from the patient, and everything in between research, basic translation, it also, the final goal is to go back to our patients. And that commitment has really structured the thinking of all of our brilliant basic and translational and more clinical researcher here at Rochester. So from very early on, we were pioneering, really figuring out that for rare diseases, we will never be able to prove that a gene therapy or the drug works in a certain disease unless we have what we call trial readiness. So, and for that, you really need to have robust natural history study, learn, how do these rare disease progresses, outcome measure, what are you going to measure to show that the medication really works, and really to be ready. So they were a pioneer in that sense. And then really a deep commitment to rare diseases. There are 7,000 rare diseases that affect very small group individually, but 30 million Americans living with rare diseases, and 8 out of 10 of these are genetic, so they are approachable by gene-based treatment and gene therapy, the majority of these also lack any FDA-approved treatment, and the vast majority are also affect children with really shortening life expectancy. So that commitment was there back in the 60s. They were thinking way ahead of all these new discoveries, and really they created the milieu and the know-how of how to move a drug to patients. And, you know, it's important as we talk about gene therapy and neurology is that, you know, here in this scheme, many of you might be familiar that to take a drug from drug discovery to FDA approval, it's a long road. And here is the scheme of how many patients it takes in clinical trials. This is obviously not, this classic path is not very applicable to rare genetic diseases and neurological diseases. So again, learning how to actually adapt this scheme of clinical trials where you can only enroll maybe 10 patients, 3 patients, but still have the scientific know-how to really prove that the drug works for that particular disease, and especially with very strong natural history data. That is something that we have learned very well. The other thing that we're very passionate about it is that once the drug, such as gene therapy, is actually FDA approved, then you open a whole different, so approval does not equal access. So one thing that we're always been very committed in Rochester is that we want to be preemptively ready way ahead of FDA approval to say, okay, how are we going to have timely and safe access to all of our patients who are candidates for this new drug, and how do we make that happen? Because obviously our system access, these are very high costly medications, they're very novel. And really, you know, how do we avoid health inequality in rare diseases, right? We have learned so much about more common diseases, and that is a big factor. And so how can we do it right for our rare diseases? So after working for decades and decades of really getting new treatment to incurable diseases, we really want to do it right. So that takes another whole set of expertise, including our ethical committees, for example, which are incredibly helpful here in our medical school. So right now we are in a time where exponentially, so this is just a short list of very good examples. Spinal muscular atrophy and Duchenne muscular dystrophy now both have approved gene therapy, but I think that we are at the verge of really putting into human trials myotonic dystrophy and many other of these diseases, which are really neurological and neuromuscular. And I want to leave you with the heart-warming picture. This is the first patient we treated here in our department, and this was December, right before Christmas 2018. This was done through compassionate use of the SMA gene therapy, and this was done because we were ready. And so while the FDA was deciding about approval for this drug, we were actually able to get the drug to you know, a few children that would have run out of age for the indication, for the commercial indication. And this is just to say that, you know, this is a team effort. It takes a village with pharmacists, nurses, and most importantly, the family and the patients who have been incredible partners in all of that we do in terms of research and clinical trials. But an incredible day for all of us who have been working in this field for a long time. So I think Dr. Lee is going to take it from here. Thank you for your attention.
Thank you. So I get to talk about how SMA changed everything and tell the story of SMA, which as a child neurologist is one that is just very fun to tell and very fulfilling. So continue on to the next slide. I'm going to start the story just by talking about what SMA is and how it presents. So SMA stands for spinal muscular atrophy. And with a few exceptions, we have 2 copies of every gene, one that we inherit from each parent. SMA is a genetic disorder, and it most commonly results from losing both copies of the SMN1 gene. So those are depicted there in the yellow boxes. So in the majority of cases, parents will be asymptomatic carriers for the disorder, and with every pregnancy have about a 25% chance of having an affected child with SMA. And interestingly, for SMA and SMN1, we have a gene known as SMN2, which is a highly homologous gene, meaning that the genetic code for SMN1 and the gene SMN2 only differ by just a few spelling changes. And even though there's over 99% similarity between the sequence, one of those changes seen in the right upper corner is that C to T change shown in green in exon 7, and it has a critical functional consequence that ends up resulting in production of a sort of smaller truncated version of the protein product, and that ultimately just gets degraded by the body. However, a small amount of fully functional protein kind of slips through and is produced by SMN2, and it helps to rescue the phenotype to some extent and sort of acts like a backup gene. And this protein product of SMN1 and SMN2, called the SMN protein or spinal motor neuron protein, it's ubiquitously expressed in the body, but the motor neurons appear to be specifically vulnerable to depletion of this protein, and it leads to what we see clinically in SMA. So not having enough of this small little protein leads to progressive weakness of the muscles. Next slide. So SMA is categorized into 5 types. I'm not going to go through all of them, but 0 is the most severe end, and type 4 is the mildest end of the spectrum. And these have been classically defined by the age of onset and the highest developmental motor milestone that a child or adult achieves. So type 1 is the most common, and type 1 is the one that I'll be talking most about today. Makes up about 50 to 60% of our patients with SMA. And these are infants who have onset of symptoms before 6 months of life. Usually they present as the floppy, weak baby. You can see in the little pictures there. And by definition, they never achieve the ability to sit independently. And without having ventilatory and feeding support, these children would die by the age of 2. And until recently, SMA was the leading genetic cause of infant death. Next slide. For type 1 SMA, what was really well established prior to any interventional treatment studies was the natural history of the disease. And so this figure is from a publication by the Pediatric Neuromuscular Clinical Research Network for SMA, for which Rochester Neurology was actually the coordination center. And it demonstrates what you see is on the y-axis, percent event-free survival, meaning alive without the need for over 16 hours of continuous ventilation. And along the x-axis horizontally is age. And what you can see is that the time points in the yellow boxes are for 50%, 25%, and 8% survival in SMA type 1. And the blue boxes denote the typical ages at which milestones are achieved for a healthy infant in comparison. SMA type 1 has historically been a fatal diagnosis. But that is very different now. And so I just want to show you how this changed with gene therapy. Next slide. Okay, so what changed? So I should mention that there are actually 3 FDA-approved genetic treatments for SMA, including one that received approval prior to gene therapy. But since today's focus is on gene therapy, here's a representation of the gene replacement product for SMA, known as onasemnogene apaparvovec. Dr. Ciafaloni has already given a lovely review of how a gene transfer construct is designed and delivered. So I'm not going to spend too much time on that, but it's worth pointing out some of the specific design elements of the gene replacement construct for SMA. So it uses an AAV9 capsid, which is non-pathogenic to humans, to carry the gene. And because SMN1, the gene is small enough we can actually package the full copy of the gene inside of there. And it crosses the blood-brain barrier and into the spinal cord, which is where we need it to go. And it's also designed not to integrate into the genome, which thereby, decreases the risk of carcinogenicity. And then lastly, the promoter that's used in this construct activates the transgene, allowing it for presumably continuous and sustained SMN protein expression, so that we really, hopefully, theoretically, will only have to dose this one time. Next slide. So here is the figure from the New England Journal of Medicine publication. It was an open label single dose gene replacement study that included 15 children with type 1 SMA, which again is clinically defined as onset before 6 months of age, never achieve sitting, and death before age 2. And what this figure shows, each horizontal line represents a patient, and their colored line starts at the time of infusion, and the green vertical lines indicate what we know to be the survival rates from the natural history studies that we have. And all 15 children survived past 20 months without the need for ventilation, which again is at the 8% survival age based on natural history is 20 months. But not only that, many of them thrived. And so I'm going to show you a video of patient 10 from this study. And as the video plays, what you can hopefully see is an incredibly cute child hanging from monkey bars. In the next video, he's just walking up to an elevator. He has this little briefcase. He gets up on his tiptoes to reach that button. And these are all normal movements, achievements for a typical child, but this is something we never used to see in type 1 SMA. Next slide. And then in subsequent studies, we further learned that early treatment is ideal. So this study enrolled patients predicted to have type 1 SMA, but they enrolled them pre-symptomatically. And again, all of the children survived, but you can see that they not only reached the milestone of sitting, which SMA type 1 babies typically didn't do, and that's indicated in the yellow box, in the yellow circles, but the majority also stood and walked before 20 months of age, the age at which we previously had only 8% survival in this population. So next slide. This ultimately led to the expansion of newborn screening because we knew that we needed to catch these babies pre-symptomatically and as early as possible. So in 2018, only 6 states had implemented statewide screening, but now we are screening in 48 states. That covers about 99% of the babies born in the United States. And in New York State, the referrals goes directly to one of the state's designated neuromuscular specialty care centers, of which Rochester is one. Those are designated by those little stars on the map, and the little red regions are zip codes from which we have of patients that we care for. So we have a pretty large catchment area. We did, with our newborn screen program, we did take a look at the first 3 years. And during those first 3 years, we screened about 650,000 newborns. We identified 34 with SMA. And what we were able to do is that we were able to see them by a neuromuscular specialist within 1 to 2 days of a positive screen. And they were treated in essentially the first month of life. And next slide. So here are some videos of one of my patients. He was identified by newborn screen. He was treated here at the University of Rochester at 14 days of life, so 2 weeks of life. He has 2 copies of that backup gene, the SMN2, so it's predicted to have type 1 SMA. And there he is rolling at 8 months, getting himself to sitting at 15 months. You'll see him pushing a Costco cart at 18 months of age. It's even full of water. And then on the next slide, I have a couple more videos of him crawling, which he achieved at about 20 months. He pulls to stand in the next video. And then most recently at about 27 months of age, he was in our clinic walking around holding onto mom's hand. And it really has been so wonderful to be a part of these families' journeys as their doctor, right? Receiving a milestone video from a parent will just brighten your day. It's one of my favorite things. But the other thing that I recognize, and maybe some of you have as well from these videos, is that this boy does have some weakness. And while we have made remarkable progress, we don't consider gene replacement for SMA to be a cure. So our work isn't complete, but we have dramatically altered the course of this disease. And we've gone from the old model where we made the diagnosis clinically. There was often an element of diagnostic delay, and without any disease modifying treatments, our management was focused on supportive and palliative care, to a new model, which is early and ideally pre-symptomatic identification through newborn screening, and then expeditious delivery of treatment, which of course is not without risk and side effects and the complexities that come with high cost drugs, but also an opportunity for new learning, new infrastructure, and collaboration. So with that, I'm going to hand this off to Dr. Sam McKenzie to talk about Duchenne muscular dystrophy and gene therapy for that disorder.
Thank you very much. All right, I have the pleasure of bringing us home. And as Dr. Ciafaloni and Dr. Lee set me up, we're going to close with the more recent FDA approval of a gene therapy for Duchenne muscular dystrophy, or DMD. So just a quick overview of the disease. Many of you might be familiar with this already, but the clinical symptoms of DMD typically show up in early childhood. There's a caveat that if we look back in retrospect, some of those signs do show up a little bit earlier. But commonly boys are diagnosed around age 3 or 4 after they have some motor delay. So you can see on the left, there's a series of symptoms that kind of go with that gross motor impairment. And then in the video on the right, this is a young child trying to get up from the floor. And you'll notice that he's really relying on his arms to sort of push up. And that's because there's a lot of proximal weakness or weakness in the upper legs in this condition. That's one of the earlier features of the disease. You can go to the next slide. So as I said, this disease isn't limited to early childhood. And if we look back, we can see there's some signs of gross motor delay really from birth, because we know that the muscle damage that comes with Duchenne muscular dystrophy occurs in utero at this point from a lot of studies that have been done. So the other thing I want to draw your attention to is sort of that period after the early childhood period. So if you look here, the 8 to 11-year-old category in orange on the bottom, this is when that motor impairment, that weakness really starts to take a toll on the quality of life for these children. And they're unable to walk long distances. That might mean that they can't attend a field trip very easily if their school is leaving the classroom for the day. They might have a hard time negotiating stairs. So typically this is the time when children will start using a wheelchair at times of the day. And usually by the early teenage years, they lose the ability to walk completely, which obviously has profound impact for a child who should otherwise be really starting to engage with peers. And into the teenage years, the disease, because we have muscles in our body that are not limited to the skeletal muscles, you can start to see some diaphragm weakness, some weakness of those accessory muscles of breathing, and children might need support breathing at night. And then finally, the typical leading cause of death is cardiopulmonary failure, secondary to heart involvement, because the heart is also a muscle. So these patients will usually die in the second to third decade of life. Go to the next slide. And just to highlight sort of the broader disease burden here, as terrible as this disease is for an individual patient, every year, in the United States alone, 400 boys are losing ambulation, coming off their feet, being confined to life in a wheelchair. And similar numbers in terms of deaths per year, 400 people per year. I haven't talked to you about the genetics of DMD, sort of on purpose, but it's really a spectacular scientific feat that we were able to get a FDA-approved gene therapy, considering some of the specific details of this gene. So it is actually the largest protein in the human body when we talk about dystrophin. And the way I like to describe dystrophin to families, it's sort of like a shock absorber. And what it does is it anchors inside the muscle cell to the extracellular matrix outside of the cell. So you have these two really important domains on either end that really tether the muscle fiber in place. And then in the middle, we have a long kind of shock absorber area called the rod domain. And this gene by itself is way too big to fit into one of those AAV viral vectors that Dr. Ciafaloni talked about. And so what people have done is that they looked at a patient who had a different form of this condition called Becker muscular dystrophy, where they still retain those anchoring parts but have a shorter rod domain. And there was a specific patient who was walking into his 60s where people started to say, well, that's really interesting. There might be parts of this protein that are less important for maintaining muscle function. And what they did from that is they sort of reverse engineered a construct that actually would fit into an AAV, taking only those critical regions of the gene. And you can go to the next slide. And so this has been a huge scientific achievement and has generated a lot of interest from biotech companies. And you can see 4 different constructs that are currently in development, the top one being furthest along, that was most recently given accelerated approval by the FDA in June. And you can see that these have kind of variations on a theme. And so just to reinforce a slide that you've seen already, the 3 major components here are the promoter, which just tells the cell, in this specific type of cell or this specific tissue, I want this gene to be turned on. So you can see all of these companies are using slightly different promoters, but the common thread is that they're all active in skeletal muscle and heart and diaphragm, which are, as I showed you, the key pieces that we want to think about in terms of a therapy. And then the transgene itself, which is that part of the construct that codes the dystrophin protein, also all different, but you can see that there are similarities on either side of the gene, which are those anchoring areas between the muscle fiber and then the extracellular compartment. And then in terms of the AAV serotypes, there's a lot of use of AAV9. One company is using AAV8, and then the agent that was recently approved uses a different AAV called AAV-RH74. And I think we can go to the next slide with that. And so here's the one that was approved. And just to show you, it uses a MHCK7 promoter, which is all based on a lot of work in the lab that shows that there's good expression in muscle and heart. And then people have looked at this gene in mouse models of muscular dystrophy, in canine models of muscular dystrophy, and non-human primates, and we show that there's good expression of this gene. And then the most important thing, anytime we're talking about gene therapy, is making sure it's safe and making sure that people aren't having adverse events that might be life-limiting or even causing death in extreme cases. So a lot of work goes into the approval, and it's not just the clinical trials themselves. And next slide. And just to kind of come back to the historical perspective that Emma started the talk with, the dystrophin gene was isolated back in 1988. And I think at that time, people were optimistic that we would have a treatment, in the next decade. And if we just think back to 1988 from where we are in 2023, it's sobering to think that it's taken, almost 40 years to have an FDA-approved gene therapy product. but I also think it speaks to the amount of work that's happened and what a great achievement it is. And if you recall the video of the child with the Gower sign, I just wanted to illustrate, as Dr. Lee did, some of these videos, really do speak 1,000 words. This is a young child who is 7, and you can see that he has no Gower sign whatsoever. So he really just jumps up off the floor, which is really atypical to see in this condition. This was one of the boys that was dosed in the phase 2 trial for this particular drug. And here's our team with the first patient that we dosed as part of the phase 3 trial, which is still ongoing. And just to brag about University of Rochester, since that's the theme of the day, we were the second highest enrolling site in the world for this trial. And next slide. All right, and I'm going to close. Not so much, I don't want this to be a sobering slide, but I think it tells us that there's work to be done. So we've really talked to you today about 2 conditions, spinal muscular atrophy and Duchenne muscular dystrophy. But I just wanted to highlight here the work that still needs to be done. But I think it's also a slide that shows us that there's room for optimism for a lot of these rare diseases. So just to show you the prevalence of rare disease, 7,000 or more rare diseases worldwide, the vast majority of which have some sort of genetic underpinning. And if we were to put all the patients with rare disease together and form a single country, they would be the 3rd most populous country in the world. So I think, even though we're talking about rare disease and these conditions can feel so isolating to families, I think there's power in numbers, and it tells us that when we work together and we work on these platforms that could be expanded to multiple conditions, there's really cause for a lot of hope. Next slide. And so if we think about the low-hanging fruit, where do we start, right? There's 7,000 conditions. We can't come up with gene therapies in the next 5 years for all of them. But we start to look at some of these common themes. What's most likely to be successful? And if we can think about a small gene, one that fits within the current vectors that we have available, maybe we need to think about different vectors that could fit larger genes, and people are doing work on that. We think about conditions that have what's called a loss of function. So if you're not able to code a protein that has some function in the cell, that's what we're talking about. And those conditions seem to be kind of the easy targets, at least initially, for gene therapy. We want to think about conditions where if we don't intervene, there are severe consequences for those patients. So I've shown you 2 conditions with SMA and Duchenne where that's the case. So as a child neurologist, I'm really focused on conditions that are impairing and cause death in kids, to be quite frank. And then we want to think about rare diseases, but not so rare where there wouldn't be, the other side of this is there's ideally going to be some commercial interest from companies to develop these things, because unfortunately clinical trials aren't cheap. So there's two ways we can bring these therapies to patients. One is through philanthropic, academically driven small trials, and then one is through getting that commercial interest to take a drug to the finish line through a phase 3 and a phase 4 trial. And next, and just to give a shout out to one of my favorite diseases. It's a horrible condition, but I think it's one of these that's a proof of concept for me in terms of developing a gene therapy is TANGO2 deficiency disorder, where these children have misspellings in both copies of this gene called TANGO2. Fitting these criteria, small gene, loss of function, about 1/3 of these patients will die in early childhood. So this is one of the missions of my basic science lab in terms of developing models of this disease using patient cells, nematodes, mice, and then also doing the work on the clinical research side in terms of better understanding this disease. You can go to the next slide. And this is my last slide. I just wanted to close with some sense of what it takes to get to where we are. So we kind of talked a lot about these two drugs that have been approved and the excellent outcomes that we are seeing and are hopeful of seeing in these patients. But really all the work that goes into developing a gene therapy is not limited to the clinical trial alone. So we need to first understand what this disease is. If we're talking about a rare disease, there's oftentimes, these are understudied conditions where it's very hard to bring subjects in, conduct these natural history studies, which is just another way of saying, what does this disease do over time in different patients? We need to think about outcome measures and how we would measure the efficacy of a given therapeutic agent and do that in an unbiased way. And then we need to think about safety and then the clinical trials. And then even after approval, what's the long-term efficacy? What's the long-term safety profile of these agents? So this is really a massive effort. And even once we have one of these treatments available, then we're kind of rewriting what's the new natural history of this disease. And that's a lot of work, but I think we're all willing to do that work if it means helping these kids. So I think I'd like to close with that. And our last slide has our contact information. And I know I speak for all of us when I say we'd love to hear from Rochester alumni and talk more about this condition. And we can certainly take any questions that have come in.
So I think that we're looking at some of the questions, but I know that we, Sam, maybe this is good for you. There was a question that came earlier on, but it's also now is about, NF1 and NF2, how easy or hard than any activity on that particular disease. I mean, you can explain what are some of the complexity of that.
Yeah, and Bo might want to chime in as well. But with these conditions, I think we've highlighted one of the major limitations with our technology currently is the carrying capacity of these viral vectors. So I think of these as like a Trojan horse, right? We are borrowing something from biology to carry this gene into the cell. And I know specifically for NF1, for example, it's almost as big a gene as dystrophin. I couldn't tell you the actual gene size, but we're more than 10 times an order of magnitude more than we could fit into an AAV vector. Not to say that that's not something people are pursuing on the basic science side. So I don't have a specific answer for those conditions in terms of where the science is. But I think there's even room for optimism with these large genes because people are working on other ways to get genes into the body, including lipid nanoparticles, non-viral vectors. And I wouldn't be surprised if in 15, 20 years, those are kind of the new thing. And we're looking back and saying, I can't believe we use these AAVs.
Yeah, there's a lot of new strategies being studied. And I think that there is a question about our patient registry for specific condition being used. And I think that goes also with another question we received about what helps with natural history study and interventional studies in which there are no approved therapy. I think the registry issue is hugely important. And in fact, at Rochester, we have been pioneer. We have a large NIH-funded registry, for example, for myotonic dystrophy and FSH1. But I think the registry before and after approval of new medication are incredibly important. And I think they really belongs to academia, right? Sometimes getting funding for those is a bit difficult, but I think that is owning the data of this transformative therapy and how the field is changing really stay in academia. But I think that what helps is really that all of these successes really takes a village, and the village is really most importantly the family and the patients. And I think that if you have a center that make a strong case and say that patients are partner in this and what we do. And it starts with natural history and with registry, which might be a lot of work showing us, so and say, okay, we're not quite prime time for a treatment, but this is what it takes once we have a treatment to try in human. So it's a long journey, it's a long partnership, but I think that in rare diseases, it pays off. And if you do it right, we have found that in our patients and family are so willing to be part of solving the problem. And we've been very fortunate in Rochester because we have started the partnership very, very early, back in the 60s. So. But do you see any other questions?
There was a question about is the near future of gene therapy and neurologic conditions mostly in the neuromuscular and muscular dystrophy space? What about other neurologic conditions like genetic forms of Parkinson's, dementias, cerebral palsy, and et cetera? And I think that now we are in a time where gene therapy is going to likely have very broad and wide applications, but the stage at which gene therapy is in study and certainly moving towards clinical use is very, very variable. It happens to be that in the neuromuscular disease space, we were lucky enough to be at the forefront of clinical gene therapy. I do think it's coming down the road for many other applications. I can't speak specifically to, I don't know all of the early sort of bench side research in all of the different genetic disorders, but just like Dr. McKenzie was saying, there are certain genetic disorders that really are primed to be very good candidates for gene therapy, and those are the ones that are probably most likely to come sooner. There's another question about what our thoughts were about why there's not full recovery for SMA patients being treated so early. And this is sort of a two-part question. And about whether there are pros and cons of using gene therapy versus CRISPR, and could it be CRISPR used as a future therapy for SMA or Duchenne disease, as it has for others?
I can take a crack at the CRISPR question.
Yeah.
And I think CRISPR is an incredibly exciting technology. I can tell you, as somebody who uses it in the lab and still has trouble getting that delivered to all the cells, even that I'm treating in a dish or in a worm, it's definitely not something where we can count on it getting to all the cells that we need, doing the edits that we need, and doing those safely. So as people may or may not know on this call, CRISPR is like little scissors that can cut out or put in nucleotides in our own DNA, which is very different than what we've been talking about, where we're adding a gene that does not integrate into our genome. So anytime you're talking about editing the genome, you have to really make sure that you're not editing in places where you don't want to edit. And I think that's been one of the main holdups. But the other holdup, honestly, is just getting it to where it needs to go and making sure that we're doing those edits in every single cell that we need to. So more to come. I think maybe we'll give the talk again in 10 years and we'll be using CRISPR in a phase 3 trial, but certainly not right now for in vivo use.
A question about, now that we have these gene therapies available and diagnostic, how much faster, right, the discovery? I think that the enthusiasm after receiving approval for spinal muscular atrophy, I think when you have the first win in a dramatically different therapeutic approach, like really gene transfer, when you have the first successful, I think that sends an incredible encouraging signal to the whole community of people that have been working on this. So I think that the field is moving much faster. So I think that a win in neuromuscular diseases is a win for the whole field. So for other, Parkinson's disease, other diseases, and it's gonna keep the momentum So I think it's positive for everyone. And also the sharing of the learned lessons across field of expertise, adults, pediatric, neurology, hematology, is the all field benefits. But yeah, we're going definitely in the right direction. Like I said, 5 approved gene therapy in 2022. So it's really picking up fast.
Real quick on the Tay-Sachs, yes, I think there were 2 kids dosed last year with that particular gene. And then Bo, do you want to close with the in utero treatment question?
Yeah, I was just typing a response, but there's no clinical trial on this right now, but it's an area of significant clinical interest, either treating pregnant moms or treating in utero. There are other disorders where we have seen in utero treatments, fetal treatment. So this is coming. There's not active clinical trial, but yes, these are all very exciting areas of research.
And any questions that we didn't get to, as I said, we're happy to take emails. We love talking about this. And this has been a real pleasure for all of us to get the chance to interface with some Rochester alumni. So
please, reach out to us. Thank you for joining and for your thoughtful question. We're available, and thank you very much.
Thank you. So we are going to wrap up now and do closing remarks. I just want to give another special thanks to our panelists, Dr. Ciaffelloni, Dr. Lee, and Dr. McKenzie for taking your time to impart your knowledge and share your work share information about your life-changing work, as well as the discussing future developments for gene therapy. I've certainly learned a lot. And as an educator, I will be bringing all this new knowledge back to my nurse practitioner students. In addition, as an advocate in other areas of maternal health, I also appreciate the expansion of the screening, because we know early intervention is so significant. Catching things early so that people can optimize health outcomes for the long run. So, some of us on this presentation may not be directly involved, but you can be involved with advocacy outside of the clinical aspect of gene therapy. So thank you again, amazing presentation. And we definitely look forward to hearing your advancements in the future. So we want to thank everybody for joining us today on Experience Rochester. And we hope you enjoyed the program, and we would like to also hear your feedback. So please take a moment to share your thoughts about the session and through a brief anonymous survey. The link for the survey will appear on your screen at the end of the webinar, and you can just click on it and go directly to the survey. Also, if you haven't already done so, please also mark your calendars for Meliora Weekend, which is going to be October 5th through 8th, 2023. And plans are already underway for this wonderful weekend of reconnecting and celebrating and also expanding our minds on campus. The headliners will be announced soon, and you don't want to miss this exciting Meliora Weekend. So registration will open up on August 9th, and then more information will be made available on the website at rochester.edu/Meliora Weekend. And lastly, again, thank you for joining us, and thank you to our panelists. Have a great afternoon and a great weekend, everyone.